Okay, welcome everyone to this webinar. We will talk today about the genomic analysis of SARS-CoV-2 new variants in this time when the vaccine is starting to be administered all over the world. And so what we want to do is we want to just discuss some of the variants that are appearing and what are some of the biological factors behind this. And first of all, let me introduce our first speaker. So Dr. Gus Kasulis, who is the head of Department of Pathobiological Sciences, and he's also the director of the Division of Biotechnology and Molecular Medicine, Biomed, at the School of Veterinary Medicine at the Louisiana State University in Baton Rouge in Louisiana. And he is also the principal investigator of Louisiana Biomedical Research Network, LBRN, and the president of the National Association of IDEA Principal Investigators, NAPI. So Gus, I will pass it over to you.
Okay, thank you, Elia. Let me share my screen. Okay, I hope everybody sees my screen right now. Elia, let me know if there's an issue.
Yeah, we can see your screen, but we don't see the full slide. We see, I think, the presenter view.
Oh, this is potentially an issue that we never resolved here. Let's see. If I put it on the presentation mode, that's where it goes. So I just wonder if there's a trick that you know.
Yeah, down there. Yeah, down there. If you, I think you see. No, go back to that screen real quick.
Okay.
In the presenter mode. In the presenter mode. And on top, you see you have end show, tips, and then swap displays. So click on swap displays. It's on the top left corner. Yep, swap displays. There we go. Now we can see the right view.
You did it? Okay. All right. That's good to know. Okay. Well, thank you everybody for attending. Thank you, Laia, for the introduction. What we're going to try to do today is give you a glimpse, perhaps, on what is going on now with all these new variants that have been appearing worldwide. And we hope that this introduction is sort of an introduction of a couple of different things that we're doing with the Louisiana Biomedical Research Network, as well as with LSU. We have formulated the graduate course, PBS 7003, Section 2, that will be starting somewhere mid-February, concentrating on analysis tools, bioinformatic tools, for looking at sequences from simple alignments all the way to structural prediction and looking at some of the topics that we'll be discussing today. Also, in addition, in collaboration with Pine Biotech and Lai Brodsky, that was our organizer for this webinar, we're going to be offering a cadre of different bioinformatics courses that will be virtually delivered through the LBRN network, and it will be underwritten by LBRN for those that belong to the LBRN network. And we hope that Pine Biotech will also extend the discount for others that may be relating indirectly to our network, one way or the other. So I want to do a brief introduction to the topic. Some time ago, I gave a full-hour lecture on the molecular and cellular biology of SARS-CoV-2 and coronaviruses specifically, and that lecture has been taped. It was actually, in fact, 2-hour lecture, because we had my lecture up front, and Eli Brodsky gave the second part of the lecture, where we did a very detailed analysis of the molecular cell biology of SARS-CoV-2, and that lecture is taped and is available I believe, on the LBRN network for those that may want to hear more of the specifics of the molecular cell biology and other tools that have been used to analyze those genomes and the structures of the predicted structures of different proteins. So as an introduction today, what I want to do is focus specifically and highlight the possibility, in fact, the actuality that coronaviruses are really more error prone than people think, because there's a lot of misunderstanding discussing the fact that the RNA-dependent RNA polymerase that's important for the replication of the virus has proofreading activity. Therefore, the genomes shouldn't really be that variant. But in fact, as you'll see, and we'll be discussing, that this coronavirus exists as a swarm, a quasi-species, a complex set of different types of virus strains that could then be selected based on immune selection or antiviral therapy and so on. So what do we see? And the take-home message is what we see now as these emerging variants is not really a surprise. It's just basically the interaction of the virus with the host, perhaps even the additional pressure that we're putting on these quasi-species with these vaccines, that ultimately selected variants would be selected, replicating at higher level, and we can see either increased pathogenicity or hopefully in the long run, a decreased pathogenicity as the virus evolves and adapts to the host. So in brief then, I've done that introduction in the past. I just want to remind you that corona SARS viruses are not particularly new to us right now. We had the severe acute respiratory syndrome back in 2002, also from China originated, belonged to the beta coronavirus group. It was also transmitted from bats to civets, and civets was the intermediate host to humans. We had an outbreak, more than 8,000 cases, 774 deaths, and a fatality rate of 9.6%. So this virus was quite lethal, but it did not spread very well. And that has to do with the receptors that the virus was using at the time. So it was very quickly contained, and we didn't have major epidemics and pandemics. We also have the cousin virus, the Middle East respiratory syndrome virus, MERS, another coronavirus, is 2012, originated in Saudi Arabia. Again, the same group of viruses. It was transmitted from camels to humans, more than 2,400 cases, 858 deaths, and a fatality rate of 34.4%. Very lethal virus. Again, it was very quickly isolated because the transmissions were also only nosocomial in hospital settings, and they were very easily contained because the virus was not easily transmissible. And now we have the 2019 coronavirus, SARS-CoV-2, that appeared in Wuhan, China, and it's the 7th coronavirus found to cause illness in humans. Mind you, we have 4 other human coronaviruses that are really seasonal. So every year, 15 to 20% of respiratory infections are caused by these already pre-existing coronaviruses. In fact, there is a hypothesis that perhaps exposure to these viruses may, in fact, prevent subsequent, at least lethal infection, but that has not been proven yet. So I wanted to also tell you that not only, as we will discuss, that the replication machinery of these viruses create quasi-species and variants that could be ultimately selected, but the actual pool of those viruses in bats, and this is the different types of viruses we've seen from different vectors, and the coronaviruses have originated from bats and the SARS-CoV-2. There are over 400 genotypes of SARS-CoV related viruses in bats right now that have been cataloged mainly from China. And we don't know what other type of coronavirus exists in bats elsewhere. So there is not only ability of the virus to alter itself during its replication cycle, but also the fact that there is a plethora of these types of viruses, different genotypes already pre-existing, giving the possibility that any of these viruses could emerge to be a pathogenic virus in humans. We also know a lot about coronaviruses from other coronaviruses that affect humans. And you can see here a number of different coronaviruses on the older category of different groups, 1, 2, 3, alpha, beta, gamma, and ultimately the Delta group. And you have the human coronavirus 229E in the alpha group, but we have also transmissible gastroenteroviral virus, porcine coronavirus, the feline infectious peritonitis virus, very lethal in cats, and canine coronavirus and others. And of course, under the beta group, we have the mouse hepatitis virus. The mouse hepatitis virus is actually a coronavirus that very accurately depicts the symptoms and disease patterns of human SARS-CoV-2 virus in mice. In fact, there is a very nice model for studying the immunopathobiology of coronaviruses in the mouse system. And and these viruses have been extensively studied. Their tropism, most of them are respiratory, but also enteric. They can alter between respiratory and enteric, as we see also for SARS-CoV-2, as you're familiar with from all the news that have and the publications that have transpired so far. So again, SARS-CoV-2, the new virus, it affects humans. There's some indications that may affect certain animals, but not really at the level where it would be used for transmission. And then we have respiratory, mostly infection, but also enteric and multi-organ infection at some point, including neurological tissues and neurological symptoms that are created because of SARS-CoV-2 infection. So in our lab, we worked also on bovine coronaviruses, respiratory coronaviruses, and that's where in a number of papers in the early 2000s, late 90s, we described the quasi-species, and we really discussed the fact that the unique replication and transcriptional strategies of coronaviruses may be causing in vivo generation of viral quasi-species, that is, these variants that exist and can be selected upon different pressure. In fact, for bovine coronaviruses, there are distinct genotypes that affect the mostly respiratory or mostly enteric, and most likely the hypothesis for us at that point was that the acute respiratory disease syndrome in cattle was really caused by bovine respiratory coronavirus that then was followed by Pasteurella hemolytic infection, which is a bacterial infection. And this is exactly what we see now in SARS-CoV-2 infections. So if we look very briefly on the life cycle of the coronaviruses, the virus is an envelope virus. It has glycoprotein, the major spike glycoprotein on the surface. This is the one that really binds the receptors. At different receptors really determines the host range of the virus. The virus then will enter through endocytosis. It will fuse out of the endosome, and then it will be ready for replication. The replication of the plas-strand RNA, so the RNA is an mRNA polarity, which is actually capped in poly 8 at the very end. And it goes through a negative sense anti-genome, replicative intermediate, and then from there on you have subgenomic messages. All those really use a negative DNA intermediate to produce subgenomic mRNAs, and this is one of the mechanisms where mutations are created. In fact, the level of variance at the mRNA replicative intermediates is much higher than what you see on the viral genome, because by the time they replicate and embed it into the genome, there's proofreading activity and stability selection to select a virus that will be stable in terms of replication properties. So this is for mouse hepatitis virus, but out of virology textbook. And you can see the fact that you have up to 31,526 nucleotides. They typically, and I'll discuss later for SARS-CoV-2, you have approximately 14 to 15 different operating frames coding for over 27 different proteins. And that enhanced protein content is because of proteolytic cleavage. The virus codes its own protease and prot here that process different proteins to create more active functional subsequences, subproteins. And also it uses, as I said earlier, the different types of RNAs that are 5' 3' co-terminal, having the poly A tail and the 5' M of the genome sequence. And this is done through a discontinuous replication where basically during the negative intermediate, the polymerase jumps at the end of that sequence through regulatory sequences and adds the 5' sequence. And that specific phenomenon is actually error prone. And This is the 2019 viral genome for SARS-CoV-2. Again, as I said, 14 operating frames, 27 different proteins. Another mechanism to increase the protein content is ribosomal shifting, slipping, where basically during the polymerase here, you could have a ribosomal shift and produce different types of proteins. The major spike protein is the fusogenic protein that determines host range because it has the binding domain for different types of receptors. But also importantly enough, the virus encodes a number of different other proteins, non-structural proteins. Some of them are structural, like the E protein and M protein and N, others are non-structural and have important and yet mostly unknown functions regulating innate immune response. And that's also pretty important because some of the variants that we see are not only in receptor binding or determining host range, but some of the nucleotide and amino acid changes may be in some of these proteins that relate to how the virus controls the innate immune response and the subsequent downstream adaptive immune response. So I think Elia would elaborate further this importance of the spike protein. The spike protein is the only fusogenic protein that determines the viral entry, binding onto receptors and fusion. We know that the S1 domain that contains the receptor binding domain determines tissue tropism, and it's sensitive to different pH conformations. In order for these fusogenic proteins, the type 1 class 1 viral fusion proteins to exert their membrane fusion ability, they have to really open up, exposing a fusion peptide, which then intercalates in the opposed membrane, creates a pore function where the trimer assembles to create that pore, and that results in fusion of the viral envelope with cellular membranes during exocytosis from the endosome, or in certain other viruses during entry at the plasma membrane. And of course, the spike protein is the major immunogenic protein, the antigen, and this is used extensively in the vaccines that we'll discuss briefly here. Then again, what I mentioned as a mechanism for entry, you have the viral spike protein attaching to a receptor, potentially other co-receptors. Then you have a conformational change and ultimately exposes the fusion peptide here in red to the opposed membrane. And then the trimer is reassembled in the opposed membrane to create the pore to mediate the membrane fusion phenomenon that results in the viral capsid freeing into the cytoplasm of infected cells. Again, in terms of receptors, I mentioned the fact that we know a lot about the different receptors. We know that some of these viruses, transmission gastroenterovirus, uses the porcine APN molecule. Different other molecules have actually used that too. But we also have the carcinoembryonic antigen M1 for mass hepatitis virus would be well characterized, as well as as a co-receptor, the O-linked acetyl glucanate. So these receptors, the binding to these different receptors, really, as I said, the host range of the virus. And of course, for SARS-CoV-2, we know now that it uses the angiotensin 2 H2 receptor. And in fact, it contains additional elements for efficient proteolytic cleavage and exposure of the fusion peptide. That's why this virus is quite efficient in infection as well as spread. And we know that from our animal studies, the deletions of the spike protein can change, or amino acids can change virulence and tissue tropism. One specific example is transmissible gastroenteritis virus, which is really an enteric, very high pathogenic enteric disease in piglets. But if you delete 225 amino acids from the amino terminals from this S1 amino terminal portion of spike, you create a porcine coronavirus, which only causes mild respiratory disease of pigs and some interstitial pneumonia. So we know that the spike protein, its receptor binding ability, and its fusogenic functions are very important in terms of its pathogenicity. We also, I want to highlight these two papers that we published back in 2005 and 2007 for SARS-CoV-1 with Chad Petit, who is now an associate professor at the University of Alabama at Birmingham. And the only thing I wanted to tell you is that we found that not only mutations, as would you'd expect, in the extracellular portion of spike, but also mutations of the carboxy terminus of the spike glycoprotein would affect the fusogenic ability and binding and functions of the receptor. So it's not necessarily that you have mutations only in the binding or in perhaps fusion peptide, but they could be distal and through conformational changes alter the function of the protein, altering its host range and perhaps its ability to infect efficiently and spread. We recently, in collaboration with Dr. Siddharama Joyce at ULM, we published this paper, and I borrowed one figure from that. What we did, we took the spike protein of the SARS-CoV-1, and which is called SARS old, and the spike protein of the SARS-CoV-2 nuum, and we expressed it transiently in African green monkey cells, and then we stained them for expression. And we wanted to see if the different proteins cause different types of cell-to-cell fusion, which is an indication of the ability of the spike protein to really facilitate more efficient entry, as well as spread from cell to cell through a cell-to-cell fusion mechanism. As you can see, the old one causes limited fusion on African green monkey cells, virus cells here, although the new SARS-CoV-2 virus causes extensive fusion. And this is also a property of the virus that is not, frankly, paid much attention in the literature, because this virus not only enters more efficiently through an enhanced fusogenic ability of the spike protein, but also can spread from cell to cell by fusing adjacent cells. That is, then you could potentially have a high level of neutralizing antibody in your lung fluids, but in reality, the virus can spread from cell to cell without necessarily seeing the extracellular space. So I wanted to bring your attention again to the accessory proteins for the SARS-CoV-2. Although they have similarities to SARS-CoV-1 and SARS-CoV-2, there are also differences. And some of the pathogenic potential of SARS-CoV-2 may be relating and depending on some of these proteins that regulate the immune system. And if you look at the functions that have been predicted, and again, we don't know a lot about all these proteins, you can see that a number of these proteins relate to innate immune responses. For instance, 3A is incorporated to virion particles and upregulates NF-kappa beta, IL-8, Arantes, and so on. It has also a role in apoptosis induction and cycles arrest. 3B, we don't know if it's incorporated in the virion particle, but it has a type 1, affects type 1 interferon production and signaling inhibition. Again, 6 and so on, type interferon production. And 8B, cellular DNA synthesis, caspase independent cell death, apoptosis, and anti-apoptotic functions. So it's easy to see and project that some of these proteins would be very important in how the virus deals with the innate immune response, evading the innate immune response, and any other defense mechanism, including apoptosis, that the cells may have in order to really be successful as an infectious pathogen. So I wanted now to focus on the new variants and try to make some extrapolations as a background to what Ilya Brodsky will be talking about. And this is really from the Central Disease Control page, is practically verbatim. As we know, we have the, that's been all over in the news now, the B117 lineage, which is the variant of concern because it's being hypothesized that not necessarily more lethal, but apparently up to 50% or higher, more transmissible. And yet we don't know exactly what that means, but that's been mostly on some clinical data and anecdotal evidence as opposed to real experiments that could really be subject to peer review. The variant has a mutation in the receptor binding domain of the spike at position 501, and amino acid asparagine has been replaced with tyrosine. So this shorthand for this mutation is N51Y that you see in the literature. And the variant also has several other mutations, including a deletion of 2 amino acids that occurs spontaneously and leads to conformational changes in the spike protein, and the mutation of proline 68 to histidine via the S1 furin cleavage site. And that furin site is really processing the S1, S2 cleavage to facilitate exposure to the fusion peptide and increase fusionicity. There's also a stop codon on ORF8, and this function is unknown. The variant was estimated to have first emerged in the UK. Since December 20, there are several countries now have reported cases of B117, including the United States and Canada, first reported in Colorado and California, as well now apparently in New York. The variant is associated with increased transmissibility, more efficient, as said, rapid transmission, and there's no evidence so far to suggest that this variant has any impact on the severity of disease or vaccine efficacy. However, it's noted that now the population that's been affected increasingly are younger populations, so perhaps this increased transmission has something to do with this, that gets transmitted more efficiently into a younger patients. The second one is the B1351 lineage. This particular virus has multiple mutations in the spike protein, including these mutations indicated here in red. But unlike the B117 lineage, as detected in the UK, this variant does not contain the deletion of 6970 that I noted earlier. This was actually identified in South Africa in samples back beginning of October 20, and cases have been detected outside of South Africa. The variant was also identified in Zambia, and currently there's no evidence to suggest that this variant has any impact on disease severity. There is some evidence to indicate, though, that one of the spike protein mutations, the E4084K, may affect neutralization by some polyclonal and monoclonal antibodies. And that would be the expectation based on the fact that these viruses have gone through hosts that mount their own immune responses, not necessarily be subject to monoclonal antibody therapy or any of the vaccines. They may already been selected as immune escape variants. And the last one I will mention, there's also mentioned the CDC, is this particular P1 lineage, the mutation that's shown here. And this one is has 17 unique amino acids and 3 deletions and contains these mutations. There's no evidence to suggest that some of these mutations in the P1 variant may affect the transmissibility and antigenic profile so far. But a recent study reported on a cluster of these cases in Manaus, the largest city in the Amazon region. And in this region, it is estimated approximately 75% of the population had been infected with SARS-CoV-2 as of October 2020. And perhaps this high level of infectivity led to this variant being selected much quicker than it would be in other parts of the world. The emergence of this variant raises substantial concerns of a potential increase in transmissibility or propensity for SARS-CoV-2 reinfections of individuals because of the plethora of these mutations and deletions across the genome. They obviously, what are the potential consequences of having these variants? You could see that in red, and I'll go fairly briefly on that. The ability to spread more quickly in people. We've seen this. There's already evidence that one mutation, that the D6014-G glycine mutation confers increased ability to spread more quickly than the wild-type virus. This particular variant propagates more quickly in human respiratory epithelial cells in cell culture, outcompeting the 614D original wild-type viruses. There's also epidemiologic evidence that the 614G variant spreads more quickly than viruses without the mutation. The other consequence would be ability to cause either milder or more severe disease in people. So far, there's no evidence that these recently identified COV viruses cause more severe disease than the early ones, other than the fact that there is a strong indication or suspicion that these viruses are transmitted more efficiently. The ability to evade detection by specific diagnostic tests is of concern. Most commercial polymerase chain reaction tests have multiple targets, though, to detect the virus, as the ones that we use here for wastewater detection in our laboratory for the Spanish Rouge Parish and LSU. So basically, by having multiple sets of PCR primers, you would avoid the fact that maybe you will be unable to detect a specific variant. But that needs to be looked at every time a variant comes in, because it's possible by accident that the PCR primers that are selected really target those altered mutations, in which case you won't be able to see that. Decreased susceptibility to therapeutic agents as monoclonal antibodies. Obviously, if you had targets of monoclonal antibodies, like monoclonal antibody therapy, which President Trump was treated with, if there is that area where the single amino acid change could really knock out the monoclonal antibody therapy fairly easily. and ability to evade natural vaccine-induced immunity. And that's where basically these other open ready frames are very important, and mutations, because we really do not understand the immunopathogenesis of these viruses. So everybody's concentrating on the spike protein because the easier target, but in reality, there are many other things that we need to know of how this virus really controls the immune response and causes pathogenicity. In terms of vaccines, very quickly, I wanted to just give you a 30,000-mile view of vaccinology. We have been working on vaccines for coronaviruses quite some time, successfully, and a number of instances unsuccessfully. We have live attenuated vaccines for porcine endemic disease virus, and that's been done by serial passage of the virus 90 times or more in African green monkey cells. The infectious bronchitis virus, it has multiple serotypes, and there are vaccines for EBV right now with a similar type of mechanism. We have killed vaccines that have been used for canine coronavirus and infectious bronchitis virus, not as successfully, but at least they provide some protection. And there are vector vaccines, including adenovirus vaccines for lactogenic immunity to transmissible gastroenteritis virus, and baculovirus for the spike SMN that has been used for preventing enteric challenges. There's also passive immunization using neutralizing monoclonal antibodies, as already has been used for SARS-CoV-2, and at the antibody-dependent enhancement with feline coronavirus vaccines. So for COVID-19 vaccines that are in human trials and already been out there in the market, as you know, mRNA 1273 for Moderna is really the entire spike protein, at least the entire extracellular portion of the spike protein. It has passed the phase 3, is actually in the market. It requires two doses, 28 days apart, and it has been shown to be 94 to 95% efficacious in terms of preventing the disease onset. The mRNA BNT162 Pfizer, it has only the RBD, the receptor binding domain, linked to a fibrin neck that facilitates trimerization. And so basically it's a much smaller target in terms of vaccine, and that similarly has passed phase 3 trials, and that creates a single or two doses that administered a few weeks apart. We have a number of other vaccines that are coming into play. We have the Novio Pharmaceutical Inc. that has a DNA plasmid. We have the Janssen Pharmaceutical Companies, Johnson & Johnson has an adenovirus 5 vector vaccine, and Oxford University with AstraZeneca in collaboration with the Russian company with Sputnik, which is the vaccine, adeno vaccine. They use a simian vector to express the spike protein for vaccine purposes, and they report 50 to 90% efficacy also for this. And those are in the market already. At least the adenoviral vectors are in the market. And then again, this is something about why the dosage here. You could see that the Moderna, you have up to 100 really micrograms of per dose. That's a lot of RNA, which is pretty codon optimized and includes specific ribonucleotide nucleotides that are being placed as synthetic there to downregulate immune response recognition by TLR3, TLR4, and TLR7, because RNA in itself is very inflammatory. And in fact, in certain patients in Germany and elsewhere, were accidentally were given 5 times the dose. They had severe problems, inflammatory response, anaphylactic shock, and so on. They have different requirements, being frozen or not. And the Pfizer vaccine requires a minus 70 degrees because really it's a much smaller protein and they need to preserve the trimer, otherwise it falls apart. Although the Moderna has the entire spike that naturally forms a trimer. And those both, the Moderna has a NIH-driven mutation, and I think Lai will talk about this, that keeps the spike protein in an open conformation, exposing the RBD domain to increase ability to create neutralizing antibodies against the RBD domain. I wanted to also tell you that we know in the past for SARS-CoV-1 that neutralizing antibodies and memory B cell responses are short-lived in SARS recovered patients, in human patients. And you can see here that in some of these patients that is the cohort itself in black, patient A, patient B, that over time, in months, you can see that the immune response, the B cell response, antibody response to be waned out. And the question is, although the studies have been done to 2-3 months right now, it will be very interesting to see what the results are 6 months down the road, whether B and T cell responses are really surviving and they're not weighing out, and whether those people being vaccinated are protected in the long term. So this is what I wanted to tell you in brief. If I want to sort of quickly summarize before I recognize my staff here, is that two things. SARS-CoV genotypes exist as a plethora of genotypes in bats right now, over 400 genotypes. In addition, the replication machinery of these coronaviruses creates this quasi-species, these variants in cell culture that could be easily selected based on external stimulus and external selection, whether that would be an antiviral or a immune selection. So it's not surprising then that we could see variants evolving. These variants were not really caused by mutational rates of the RDP polymerase, although that also could play into this, but it could be also pre-existing as part of the quasi-species that are naturally selected. In fact, I think Elia will mention a recent paper where they took the virus SARS-CoV-2 and they put it in cell culture in the presence of human antibody, and they selected variants very easily that mimic some of the viruses that we've seen so far. So we are really at the beginning of understanding how these viruses evolve. I think bioinformatic analysis is extremely important because as we see these variants are coming through and maybe even detecting them ahead as part of the quasi-species, we could potentially predict the outcome of these variants, which one would persist or create a different pathogenic profile. So I want to thank you for your attendance, and I want to acknowledge people in my lab, the General Medical Virology cover and paper on nelfinavir, the difference between spike proteins for SARS-CoV-1 and 2 were worked by Dr. Fahana Musarat and Dr. Vladimir Julchenko. Also, everybody else in the lab has contributed to a lot of the work that we're doing so far, including some of our vaccine efforts that are ongoing. We have in the lab adenoviral vectors and herpes vectors that express spike protein, and we're actively involved in vaccine experiments. And of course, all our collaborators, and acknowledge the collaboration with Eli Brodsky and the Tober Bioinformatics Research Center at the University of Haifa, which is really where Elia originated, at least the company got spun off from that Tober Center. So thank you, and we can proceed then, Elia, with your presentation, unless there are some questions at this point. I don't know how you want to handle that.
Thank you, Gus. Yeah, so I imagine that some people might have questions about your presentation. So we'll just, if you have any questions, please put them in the chat. We will address them maybe towards the end of my part of the presentation, which is not going to be very long. So again, if you have any questions, and thank you guys for the presentation, please put those in the chat. What I wanted to do is kind of pick up on some of these topics and expand a little bit to talk about the bioinformatic aspect of how we can piece together this data. Where does the biology or the biological understanding of this virus intersect with the data that is available and that is being generated daily? And how do we piece together the whole puzzle to kind of think about the impact of this rollout of the vaccines and the impact of how diverse the virus will eventually become as these additional treatments are introduced and being used more widely. So as Gus already mentioned, many of the vaccines and early research on coronaviruses and SARS-CoV-2 primarily has been focused on this key role of the spike protein, which plays an important role in zoonotic transmission and in general in cell entry. And so when we look at the SARS-CoV-2 genome, you can see it's encoded right here in this region. And what it does is it has this particular property that it actually is formed by these two subunits, and those are cleaved so that the S1 subunit can attach to the membrane. So what you can see here is the significance of some of those terms that Gus was using throughout his part of the lecture, the receptor binding domain, and different parts of this spike glycoprotein. So what has happened since then is that there has been a continuation of data collected from these different studies. And so here we see an excellent example of how structurally people have started to understand the way that this protein works. And so what was done in this study is that this spike glycoprotein was captured, connected to the virion itself. And you can see that the virion actually contains both of these conformations. So you can see that in general it has two forms. The thin structure that resembles this needle-like post-fusion structure, and then the wider one, which is the pre-fusion structure. And when they captured this intact virion, about 97% were pre-fusion and about 3% were post-fusion, and about 25 in total per virion. The researchers also examined the thin and the wide trimer shapes and found that they showed a very interesting correspondence. So we don't know everything about how this works in real life, but this kind of data really allows us to see the structure intact. And so what is interesting about the structure is that it's not static. It's not always kind of protruded outward, but it actually can be found in many of these. So it's kind of shaking, right? So you can see right here that perhaps it's kind of dynamic. And so it also could be in multiple ways, either open, closed, two open, three closed, one open, right? So there's a variety of shapes that you can expect here. So to really understand how this connects with the genomic data, I think it's important to appreciate the data. The majority of the data that we have is actually genomic sequencing data. And so the genomic sequencing data, when it is pieced together with this kind of an understanding of the structure of these proteins, it really provides a lot of insight, I think, into how we can understand what's going on. So here you can see this S1 subunit on top, the S2 subunit on the bottom. And here's a little video of what happens if you detach the top part. So it's like a cap that's sitting on top. And the reason I'm showing this is because I think once we go into the specific mutations, it becomes very interesting when you think about how does this practically work. So when originally the major question was about the origin of this coronavirus, and Gus explained it in great detail, one of the major findings during that time was that the novel coronavirus was very similar to one of the bat coronaviruses that was captured not long before the human version was sequenced. And so one of the questions would be, well, what is different between bats and humans that so many different viruses that end up becoming endemic or pandemic acquire these properties? So how does that environment really play a role in what happens? And so one of the things that you see here, you probably already recognize that the majority of these mutations that are differences essentially between the rat G13 genome and the human COVID-19 genome are found in this receptor binding domain region. But also if we look overall how these relate to other bat coronaviruses and other SARS-like coronaviruses, we can see that there are very specific properties that have been acquired through this transformation. And so one of the questions that was posed in this paper was, what is so unique about bats that kind of puts them in the center of all of these pandemics. And so An et al. Provide evidence that one mechanism bats have evolved to limit excessive inflammation during viral infection is through dampened transcriptional priming and lower functional capacity of the bat inflammasome sensor, NOR family pyrin domain containing 3, which is NORP3, and it functions as a pathogen recognizing receptor to activate inflammatory mediators. So this inflammation has also been linked to aging and age-related chronic diseases. And the study provides a mechanism for and a provisional answer to whether bats have evolved to tolerate or resist viral infection. So this work really suggests that bats have adapted to tolerate viral infection without development of pathology through inflammation, which explains essentially that the virus can replicate in a bat without leaving the same host for long periods of time. In fact, I think in this paper, it was mentioned that it could be several months that the virus replicates continuously inside the bat. And this is very different from the types of hosts that do show inflammation, that have kind of acute disease. As a result of both the pressure that the virus is under and the long time that it can kind of exist in a single host makes it a unique situation where many novel mutations, recombination, and properties are acquired. So to understand some of these properties, right, we started talking about the different domains. And so one of these regions in the receptor binding domain, which you can see here on top, is really the binding to the ACE2 receptor. And so it's easy to kind of imagine how, because of this evolutionary pressure for the virus to escape this high pressure environment in the bat, find new hosts, eventually it is able to primarily kind of adapt to these novel ACE2 variants that are found in other hosts. And so what we can see in this original kind of those mutations that we were looking at, right, is that those mutations are very close to that binding. So again, this is a trimer, so you can see here 3 times the same thing is replicated and only one of the receptor binding domains is open. And so you can see the ACE2, the portion of the ACE2 that is bound to this receptor. So when we look at these mutations, they're found specifically in that portion. So now the question is, right, so this was about the origin, and this was about the question, well, how did the virus acquire the certain properties? How was it introduced into the human population? And so now we have a lot more information about what happened after that. So essentially, if we look now, this was taken yesterday, I think. So this is from NextStrain, website that displays genomic information collected from around the world and analysis of that genomic information to understand the relationship between the different clades that are emerging. And what you can see here is, first of all, how the different clades are divided up throughout the world. On the website itself, you can see a nice animation of how the viral strains kind of originated from different places. And that's essentially the goal of this analysis here on the side, is to demonstrate how individual dots, which are samples, are representative of different clades. And so you can see a phylogenetic tree. And if you see here on top, there is a total of 12 clades, right? So right here, if you just count these up, there's a total of 12 clades. Now, what's interesting is that this is showing essentially 1 year. So from January 2020 to January 2021, you can see how for a variety of reasons, really, there has been a gradual increase in the number of clearly defined clades. So you can see here we've got 3 up until about February, and then we have about 5 here until May, and then about 7 to 8 kind of emerge over this period of time because one of them kind of disappears almost. And then today we're looking at 10 kind of major clades that are known. And then here, if you actually count all of these, you can see that some of them are very minor, right? So some of them are either emerging just like we saw here. So some of them are emerging and growing in number, or some of them could be just kind of appearing and disappearing. And so the reason I'm showing this is because we now have this tremendous collection of genomic information. On one hand, it allows us to analyze the data and understand both the origin and the dynamics of this disease as it spreads. On the other hand, it is also limited due to a variety of biases. For example, we know that most of these samples are collected in areas of outbreak, right? So if there's not an outbreak, we don't have any information. A lot of the sequencing also happens around the major outbreaks. And so we do expect this data to be informative, but also some of the questions could be limited. But some information is already emerging that is going to be very interesting for us to look at today. So for example, one of the things that if we do the exact same thing on this on NextStrain website, you can zoom in and you can look at how specific proteins acquire mutations. So at what rate do they acquire mutations? So an interesting thing would be to look at the spike protein, just like we were looking in terms of the zoonotic transmission, and ask the question, well, how are these mutations distributed throughout this protein? So you can see here, this is the spike protein. So it's about 21,600 to about 25,400. That's the length of this gene. And here you can see the S1 subunit and the S2 subunit. And so it's fairly clear that the S1 subunit has many more mutations that we can observe right here. And so here we do have some mutations, but essentially the S2 subunit has less mutation. We can map these mutations onto the structure of this protein data bank ID PDB6VXX and kind of see visually that the distribution is not random. So there is some pattern, and that's really what is interesting about this, is what is the pattern and what is shaping the pattern of distribution of these mutations to understand how can similar events as we go through this pandemic, how can we kind of anticipate what's going to happen, and how can we detect this early on to be prepared to what's going to happen. So let's think about the selective pressures that this virus is undergoing, because right here you can see kind of a nice chart that was published not so long ago, where you can see the different regions, so the different genes on the genome and the synonymous and non-synonymous mutations. And you can see that ORF1A, for example, has the majority of these mutations, right? So the y-axis is the number of mutations, and these are the individual genes. So the first type of kind of a selective pressure that we know of is remdesivir, right? And just in general, antiviral. So how does remdesivir work? It's a drug that is going to interfere with the RNA polymerase, or DRP. These antivirals, essentially, they are intended to stop the ability of the virus to replicate, right? So if the virus actively replicates, remdesivir is supposed to reduce that number. But at the same time, it has kind of a negative effect. Why? Because the RDRP can mutate, right? So in order to evade this pressure, and you can see right here an example from a publication where such mutations were observed, essentially developing resistance to this antiviral. So that's one type of pressures. The second one is interferon. So because there is kind of a balance that ultimately is the most beneficial to the virus, where the host doesn't die, the host continues to live, but also the pressure is not strong enough to eliminate the viral population. So when that balance is achieved, right, that is kind of the optimal situation for the virus. But right now, there is this strong immune response. We know that the virus is actively trying to control this immune response. And so there is some optimization still in that process. And so that's another kind of theoretically evolutionary process or a pressure for the virus to achieve optimal balance with the immune response. And then the long-term immunity, right? So the antibodies is another kind of a pressure. So the evasion of this immune response of detection and kind of a long-term sustained recognition of the virus is another major pressure. So these three pressures, and we'll talk in the end of how they relate to actual vaccine rollout, are kind of strong theoretically pressures that we should observe in the virus. So let's see whether this has really been seen. So the first thing that we talked about is remdesivir. The way it happens is it actually interacts here with the RDRP. So after the virus has entered the cell and the RDRP has been produced, it blocks it. And so that replication of subgenomic transcription does not happen. And so what happens is that it blocks a particular binding pocket. And as a result, that pocket becomes the region of active mutations. And so you can see here a paper that was recently published where emerging SARS-CoV-2 mutation hotspots include a novel RNA-dependent RNA polymerase variant, so RDRP mutations. And so here you have the treated versus the untreated samples, and you can see that specifically in the treated ones, there's this kind of more narrow distribution of these mutations, which indicates that on average, there's a rate of mutation that happens throughout the whole RDRP. And then when we look at this particular pressure, it is more selective. And so this is, again, it has not necessarily been studied extensively enough for us to understand what might be the rate or at what levels of remdesivir use throughout the population these types of mutations will occur, but it still allows us to anticipate what might happen as a result of antiviral treatment. Now, another kind of an aspect that we talked about was this ability of the virus to evade detection. And one of the ways that it evades detection is by these glycan molecules, so glycosylation. Essentially, the spike protein is covered with glycans that are preventing accurate detection of the full protein. And to remain flexible enough to bind to ACE2 receptor, you can see that it actually is not covered throughout. So these are mutations that happen as a result of this changing dynamic structure of these glycans that are covering the whole kind of body of this protein. So there has also been some modeling done to kind of show us what that really looks like. And so we are used to see this spike protein like this in a naked shape. And here you can see a model, computational model, that shows what it looks like when it's covered with these glycans. And so you can see how essentially the top, this receptor binding domain is open. And so that's the critical part for detection. But then a lot of the areas are covered up with these glycans, and that helps the virus evade immune detection. And so because of this, we know that this specific receptor binding domain is so critical, not only because of the transmission, the zoonotic transmission, but also as the vaccines are being developed to target that particular domain. So the other studies that also Gus has alluded to were done with the convalescent plasma. So what is convalescent plasma? Essentially, it is plasma collected from patients that have already recovered. And so that has to do with the process of disease progression. So in the beginning, there's this acute host response, and then there's this kind of response that becomes a long-term response. And after that time, after the person recovers, that plasma could be collected, and essentially those antibodies could be used as a prophylactic or as a treatment. And so here there's a study that was done. Again, the study is not conclusive in the sense that it was not done in animal models or patients, but it has been observed. So here you can see the x-axis is time in days. So from 0 to 100 days. And here you can see how these mutations are starting to occur based on a number of kind of variations that were done in the cell culture experiment, where different dosages were administered for a long time. And so here you can see different types of mutations and deletions that come out seemingly as a response of administration of this plasma. Now, one of the things that I think it's very nice to understand as you're thinking about these mutations is what is the actual structural interaction between these antibodies and the spike protein. So there's actually a PDB structure that was published that kind of shows us exactly how that works. So here you can see this, so again, here it's showing the antigen binding fragments. So there's the light chain, and so this is the outside. And so the antibody essentially is keeping this protein inactive by blocking these receptor binding domains in a way that they cannot access and bind to the ACE2 receptor. So I think this is why it's so important for us to link the mutation data and the sequencing data with the structural component, because we can really understand how these interact. So it's really nice to have such models. So this particular antibody is called EY6A. It was retrieved from an individual convalescing from COVID-19. And what they have done in the study is they have shown that it neutralizes SARS-CoV-2 and cross-reacts with SARS-CoV-1. So it's actually working with both SARS-2 and SARS-1. It binds to the receptor binding domain of the viral spike glycoprotein tightly, and the complex identifies the highly conserved epitope away from the ACE2 receptor binding site. Residues within this footprint are key to stabilizing the pre-fusion spike. So again, how effective is this going to be? This is actually a question, right? Because the regulation of how the spike protein opens up and how different portions of it are accessible and recognizable is going to play a role in how this evolves over time. So now another example, I think, is to what Gus alluded to here about these quasi-species, or essentially how instead of what we think of one single genome in the host, essentially this is a whole population or a swarm of competing virus genomes that are essentially suppressed by this more dominant, more effective genome. And so what has been shown fairly clearly is that most mutations are cleared out. And so the genome is fairly stable, not because there are no other variants out there. The population is very diverse, but the other kind of variants that are present in that population are easily cleared out. And so one key reason for that is because the infection itself is fairly short, so that they don't stay in one host for a very long time. But as that time grows, that's where you can start observing how this population really works. And so what they've done here is actually, this is I'm going to talk about a combination of two different studies. One study was with people that have a poor immune response, and the other one is with people that have this prolonged chronic condition and eventually are treated with convalescent plasma. So here, this shows the quasi-species generation. So the wild type SARS-CoV-2, which is the most common form, serves as the reference RNA sequence for development of an assay. But we can see that this essentially represents a whole kind of, each one of these clades here represents a population essentially. And you can see here individual patients. So individual patients are going to be are going to be shown here. And you can see that some of them become outliers. And so what does it mean that they are outliers essentially by themselves? That means that they have some established strain from this population that previously has not been the dominant strain. And so when they wanted to characterize what these strains look like, they started looking at specific mutations in the spike glycoprotein, because the only way to treat these people that have this infection for a long time is to administer them with convalescent plasma from recovered patients. And so here you can see how these specific mutations, including in the RDP, RBD domain, in the fusion peptide, in the heptad repeat, right? And the more importantly, they actually correspond with the epitopes that are recognized by the different antibodies. So this is one reason why prolonged infection and seemingly effective care with multiple ways to intervene in the disease actually could also bring out, through selective pressure, some of the less dominant substrains that all of a sudden are not being captured in that treatment, right? So the treatment kind of suppresses the major dominant strains, and these other clades, these other quasi-species have more freedom to replicate and become established. And so that in itself, prolonged treatment, comorbidities, and the treatments with antivirals and convalescent plasma could lead to that effect. Now, another implication of that, and this was proposed, this is a hypothetical, is that theoretically, non-Hispanic whites and African Americans might have differences because of similar properties, right? So here we see comorbidities, longer time, different types of immune responses. And because of that, this kind of change in the balance of the dominant kind of quasi-species could result in lower detection and essentially a higher mortality rate. So this was a model that somebody proposed. Now, already Gus mentioned here that there were additional studies on cell cultures, and the objective of this study here to show here is that multiple mutations occur at the same time. And so you can see here mutations in the E and the N protein. You can see a mutation in the RDRP, and you can see multiple mutations in ORF1A. And these mutations actually happen in different patterns at different days. And so one of the studies wanted to see what is the, again, structural component of these mutated regions. And what they have discovered is that one of them is directly related to resistance to antibodies, because they examined a published spike structure and kind of annotated with the residues. And it showed that some of these mutations happen in a disordered glycosylated loop at the very tip of the right here, the NTD, and therefore could alter binding of antibodies. The other mutation right here is close to the binding site of the polyclonal antibodies derived from COV57 plasma 6 plasma. And also you can see that a lot of these mutations essentially interact with these antibodies. So what we can understand from here is, one, that the longer the disease, the more these mutations occur at multiple regions at the same time. And I think another interesting thing that you can see here is that there are not only mutations, but there are also deletions. So another thing that I think was overlooked in the literature, there's a lot of conversation about different mutations emerging and being established in these clades, but not a lot of people are looking at multi-nucleotide deletions. So not just deletions of single nucleotides, but deletions that are longer. So here you can see some examples. So in the beginning, what happened was, in this study, the researchers were trying to identify deletions that are more than a single nucleotide. And you can see here some shorter examples, but you can also see right here, this is deletion length, and this is number of events. So if you just take all of the publicly available data, what is the distribution of deletion lengths? And here you can also see that they have annotated specific recurring deletions that are more than a single nucleotide, and where they also happen, right? So I think this is very interesting because there is this kind of a naturally occurring phenomenon where whole portions of important genes could be deleted from the viruses. And essentially that is a part of this quasi-species dynamic where defective genomes that might have profound effect on how these proteins are actually produced start interacting with the wild type genomes. And so this is a part that I think is going to be more and more explored, because essentially what it shows us is that we don't even know what other members of the quasi-species are out there. This is very poorly annotated and very poorly studied. And at the rate of infection, and as we see the number of established clades grow, and as we see the number of mutations in various regions grow, what it leads us to believe is that even though the current vaccine is going to be, and there are studies ongoing right now to demonstrate whether it will be effective, it's probably going to go from very high rates of effectiveness, and that will go down. But the good side is that really these vaccines, the mRNA vaccines at least, and the vaccines that are being produced kind of using this novel technology, are also much easier to manipulate. And so continuous surveillance of these viruses, especially using deep sequencing to reveal the low frequency mutations, continuous monitoring of how this kind of happens within patients, that we start to developing a sense of what happens in different cases in different clinical scenarios, as well as ability to understand the implication, the functional implication of these different mutations, and to be able to design new versions of the vaccine, right? So I think that, most of us, at least I, had this anticipation that the vaccine will be a final solution. There will be kind of a resolve of this pandemic. And, it seems like this is not necessarily true. And there's a lot more research that is going to be needed to continue and explore how the dynamic of this virus is going to continue and change, especially after such widespread vaccination. So before we conclude and go on to the questions, I briefly wanted to mention that these examples that we've introduced today are essentially examples that we will cover in detail in the upcoming Bioinformatics for Infectious Disease graduate course at OSU, which is also going to be available for LBRN. So this is something that you can learn more about on this link, and I'm going to just paste this link in the chat box. And this program is essentially designed to introduce biologists and those that are interested in such research to be efficient with the data that's out there that is being generated, with asking questions about the data and being able to find evidence in the data itself, to understand different methods of analysis. I think what we can see from some of the examples that we presented today is that it's not just the same method that solves all the questions. Really, this is a combination of a variety of different methods, and that includes sequence alignment, that includes evolutionary studies that allow us to understand relationships between viruses, and there are a lot of different methods for that that include a variety of different questions that could be asked. It's working with the structural proteins, so all of the visuals of those proteins are very easy to produce, but they are very informative when you identify those important mutations, and also analysis of transcriptomic data. So how is the host responding? For example, if we delete a certain protein or we introduce a mutation, how can we study the results and understand the structural kind of components, and what is the functional result of those mutations? So this program is going to help you understand pathogens and the diseases that they cause. We'll talk about SARS-CoV-2, but we'll also talk about other types of pathogens, and we'll also talk about this pathogen host interaction. We'll talk about different types of analyses, but also where to find data and how to start your bioinformatic analysis from data that's available, and also what are some of the applications of such analysis. How can this analysis can help us think about the function? How can it help us think about vaccine design, antivirals, antibiotics, and how can it help us understand epidemiological trends in drug resistance? And also what's important is that we are going to actually take examples from the public domain of data sets that have been curated with publications. So a lot of the examples that I presented today, the data is available. In fact, in terms of how much data is available, there's over 3.5 million viral sequences and over 13,000 genomes out there. So there's a huge variety of data. This data is easy to find and easy to use. And importantly, I think since Ebola virus, which was really the first example when, immediately, almost in real time, so much data has become available. With COVID, because so much focus on COVID and because the availability of the sequencing technologies, that rate of sequencing and availability of data has grown many-fold. And today there are, just for SARS-CoV-2, there are over 180,000 SRA runs, which are FASTQ files with a lot of information in them, over 50,000 nucleotide records. And so a lot of this continues to kind of accelerate as we speak. Importantly, I think that these data sets can provide us with important insights. And so specifically, we'll look at examples of how to correlate genomic diversity with population, for example, geographic location. We'll talk about immune responses and how to find biomarkers. And we'll also talk about association between pathogens and the microbiome. And we'll also look at not just the nucleotide and structure, but we'll also look at pathway analysis and interpretation and things like that. We'll take a look at different types of sequencing methods and how those different sequencing methods produce different type of data, and different type of data could be analyzed using different types of resources. And we'll also introduce you to some of the typical analyses without the need to use coding. So we'll do some coding, but we'll also primarily focus on the logic of analysis. So how to ask the right question, how to find the right method for analysis, and also how can multiple methods of analysis could be combined together to really develop a sense of what's going on in these complex relationships like quasi-species and haplotypes, or the emergence of more differential mutations or less differential mutations. And how can we think about asking those right questions and answering them with the correct methods? So we have prepared a number of kind of curated projects, as I mentioned, that will explain some of these for both asynchronous and in-person sessions that we'll do on Zoom, just like this. And we'll also be able to share those for further study after that. And so an important part that I wanted to mention is that all of these program resources are going to be available not just for this graduate course, but also for the Louisiana Biomedical Research Network. So the Louisiana Biomedical Research Network will offer a lot of these resources as asynchronous resources for faculty to use and teach the students. And also to kind of explain how all of this works, we will have a special program that's going to last for from now all the way until the summer, where you can have access to 16 different hands-on asynchronous courses, the cloud research tools, some coding practical tools in R and Python, and online support from instructors. And I just wanted to make sure that we kind of cover all of these options. And in maybe the future, we'll also introduce in greater detail, specifically, what will the structure of the programs be like, and how these programs can help faculty, graduate students, as well as undergraduate students. So at this point, I'm going to pause and just see if there are any questions. So again, I will read out some of the questions that are available right here in the chat. Okay, so Therese is asking what percent of the viral genome needs to be changed, mutated for a strain to be typically considered a quasi-species? So Gus, would you like to answer that, or do you want me to handle this one?
You can take a shot at this, and I could add to this if you want, like.
Okay, so the idea behind quasi-species is to identify, so if you imagine a FASTQ file and it has a variety of reads, and we're talking about here whole genomes, is to identify groups. So it's not necessarily how many mutations, right? So specifically when we talk about quasi-species, it's the detection of those and grouping them by similarity. So what could be found is, for example, mutations that are in close proximity in one region, right? And there's a lot of them, or you can find many of them distributed throughout the genome. So it's not the number, it's more how do they form groups, because the main thing here is to differentiate between errors in sequencing and some technical variants, as opposed to actual biological representative of a genome that is potentially in this sample. Did you want to add to that, Gus?
Yeah, so basically, quasi-species is really dependent on the replication kinetics. So if you look at cell culture, what we've done with bovine corona, and Dr. Radovan Chuljenko looked at that. If you look at the level of mutations of the subgenomic messages, actually quite higher than what you see in the genome, as Elia mentioned, because they're selected out for stability purposes of the genome level. So there is a real swarm of viruses, but most of them are really on lower, they're selected out, so they're predominant variants or predominant genotypes versus minor genotypes. But because of this swarm being available, based on some selection, in cell culture, in the absence of an external selection, you're going to get a virus which would be much easier to replicate. So it may not need specific functions. So you are prone to get more deletions and for non-essential genes because the virus is optimizing its genotype to really replicate efficiently in cell culture without having to worry about immunological function. And specifically for African green monkey cells, which are interferon type 1 interferon negative, there's no even interferon involvement. So basically, you're going to see certain variants be selected because of the fact that there's no interferon around that perhaps would influence the replication. But I think the point to impress is that the virus has quasi-species, swarms of viruses, in addition to mutational rates of the RNA-dependent RNA polymerase. So it would be quite easy for new variants to be created, as Elia mentioned, based on even longer infection. So the longer the infection, the higher the chance of a variant to be selected. And this is the issue because typically in SARS-CoV-2, you may have 2 to 14 days, up to 3 weeks from primary infection to show symptoms. The virus could replicate for up to 2 or 3 weeks, and the virus may be around for months before it gets eliminated by the immune system. In which case, by the time the end result would be that the virus could be transmitted in an altered form to a subsequent person, especially from those shedders that have virus for a long time.
Okay, so Therese, hopefully, okay, yeah, so she said thank you. And I do want to mention about quasi-species because I think this is a fascinating question in terms of the vaccines, right? So as we think about this, one of the methods that I had a chance to work with was called SARS, and back then it was applied to polio and dengue viruses. But what it reveals is kind of like a fitness landscape of how the virus kind of selects the dominant genome that we essentially sequence, right? So when we sequence, the majority of the sequencing information is going to come from that dominant kind of genome. But if we had a lower detection limit, so the rate of errors would be lower, then we could pick up these lower frequency mutations. And if we do, we could study the landscape to understand which ones are more likely to be beneficial sort of for the viral replication, for the overcome of these immune pressures, etc. So I think one of the opportunities here with these novel sequencing technologies, the longer reads, the lower error rates, the novel library preparation techniques, is really an opportunity to survey the population in much greater detail and perform these experiments to understand what's the logic behind the emergence of some of these low frequency mutations that already exist in the population, right? They're not necessarily new mutations. They just become more detectable as we see the whole population kind of change because of these pressures.
And as Eli mentioned, the issue with these vaccines, especially the Pfizer vaccine, which is only the RBD domain, obviously you do have potent neutralizing antibodies created, as well as CD4, CD8 T cells, because there are epitopes there. But it's very possible that the efficacy of this vaccine will wane with time. It's not going to be as bad as flu vaccines because of genetic diversity and reassortment, but you will be variants created, and the end point would be that those vaccines could be efficacious at 50% or less, which require an adjustment on probably on a yearly basis or even earlier, having a vaccine that may then target a new genotype.
All right, great. So I know that we went a little bit over time already, so we're not going to make it too long. It's already 4:20, 4:30 almost on Friday. So thank you everyone for joining today. We will follow up. We have most of your registration information for this webinar, so we will follow up with some more information about these upcoming programs. And we will probably have another webinar about the specific program structure, right, for Louisiana Biomedical Research Network and others. And I do want to mention, I see that some people are asking here in the chat privately that we have some other similar programs that essentially are ongoing at University of Ghana and with some other universities. So we'll share that information with you by email.
And then for the LBRN network, you'll find this information on the LBRN website. Although they may not be enrolled to your actual credit course right